All right, picking up where we left off last time, again, talking about all the things that the bacteria need to grow happily. And a lot of these are things that we do for them in the lab. And one of them is that we put them on some type of media that they grow happiest at. But there are lots of different kinds of media, and we group the different types of media based on what we know is in it. So defined media, we know the exact chemical makeup. So the exact chemical makeup is known. We know exactly how many sugars, proteins. We know exactly how much of everything is in there. Media, hence its name, is a little bit more complex because we know we add a lot of stuff in there into the media, but we also know that as it sits in the media, we know it starts to break down, which is fine because some of the stuff that we put in there, we want to break down to release kind of smaller nutrients for our organisms. But that means we don't know exactly how much is broken down. We just know some of it's broken down. So complex media contains compounds from the breakdown of added nutrients. And I'll give you a couple examples. Some of the things we can add onto our media that we know will start to break down and can get broken down. We add beef protein. We know it can get broken down. There's actually things just called beef agar. We can add yeast, which can start to break down. We can add soy. Even blood itself can start to break down. So we add these things because we know even as they start to break down, they still provide nutrients for our organisms. We just don't know the exact amount that's broken down. Selective media we are going to use in lab this semester. Selective media contains an inhibitor or a group of organisms or microbes. Which means there's something in there that some group of bacteria doesn't like, which means they're not going to grow on there. And it allows us to study other organisms that will grow, because we know what organisms are inhibited by these different inhibitors. And we'll work with some of these this semester that we know inhibit just gram-positive bacteria, meaning everything that grows on the plate, gram-negative, because we only want to study the gram-negatives. Differential media, these contain a substrate that if, and it's like a big capital if, broken down, results in a color change. We can add different things to the media, and we can look to see, can the bacteria do something with the substrate or can't it? And we will easily know if they can or not, because we'll see a color change that occurred. And transport media, I think I have defined on one of my next slides, so I'm not going to even write it up. This is showing an example of selective media. So nutrient agar is kind of like our blood agars, just without the red blood cells. It grows just about anything and everything, which means there's stuff all over here. However, if I just want to know if a patient has a fungal disease, I don't care, like on their skin, they'll do a skin scraping. I don't care about the bacteria that we know is all over our skin. I don't even want that to grow on the plate because that's just going to interfere with the fungus. We have specific plates that inhibit bacteria growth, but then have no effect on fungus, so that we can then study, what it is that we want to study. Just an example, because we have this up in lab. It's known as Sauerkraut agar. It has a lower pH. Bacteria are not going to grow at 5.6, at least not happily, but fungus don't care. And so if a patient has a fungal infection, we can see that a whole lot easier than trying to figure out where is the fungus mixed in with the bacteria, and they're all competing for space or nutrients, so it's not going to be as obvious. So we have selective media to inhibit one type or group of organisms so that we can study another type or group of organisms. Blood agar, it is a nutrient agar. It's meant to really grow just about everything, but it's also a differential media, meaning there is a substrate in there that, if broken down, results in a color change. And that substance are the red blood cells. We'll talk more in lab in a couple weeks, that some bacteria can fully break down the red blood cells. And you saw this in some of your plates, depending on what you swabbed, that you could see right through the plate. That was a complete breakdown of red blood cells. And the red blood cells aren't really there anymore. I'm like, it looks yellow. They're like, it's yellow. If you held it up in front of a light, it wouldn't be yellow. Some only break it down partially, and it will actually show a greenish color to it instead of red, and some don't break it down whatsoever, and it still looks just as red. So there's a substrate that, if broken down, yes, can change colors. I don't know if, again, I have no idea if my speaker's working, but I'm afraid to also plug in my clicker. We'll work at the semester in lab. It's called a MacConkey agar, and it's cool because it's both selective and it's differential. So it has an inhibitor in there that inhibits a type of organisms, but it also has a substrate, so the ones that do still grow will or can show us color changes. So this is showing an example of a nutrient agar that's meant to just grow anything and everything. We put Staph aureus on it and E. coli. Why are those two bacteria familiar from like an hour ago? What bacteria do we work with in lab today? I'm going to say Staph aureus and E. coli. So if we put them on nutrient agar, even our blood agars, they're going to grow, which is why we gave you blood agars. However, if we put those same exact bacteria on a plate called MacConkey grow, MacConkey inhibits gram-positives. Spoiler alert. Staph aureus is gram-positive, so it is inhibited. It's not going to grow. E. coli is gram-negative. It's not inhibited. It will grow. This last plate is showing us two different bacteria. It's not the same. It still has E. coli, but we've also added a new bacteria called Salmonella enterica. It's the same bacteria that causes salmonella sickness. They are both gram-negative bacteria. They are both growing on there, but we can start to see a color change difference between them. And so if we have a plate like E. coli, or like a plate like MacConkey, we can be like, okay, something grew, it's gram-negative. Did it do a color change? We can really narrow down what that bacteria is just based on, what the gram stain result is without even gram staining. And we already know a little bit more about it, whether it can break down the specific sugar we added in there. Now, not all of our media will be on plates. Some of it will be in tubes. But yes, we will add bacteria to tubes, and it also has substrates in it. And if it can break it down, you may get a color change. A lot of times the color is changed just because the pH changes. We've got lots of pH indicators that change colors. And then, I didn't write it down there, but transport media is another group of media. This media is not meant to grow the bacteria. It's just really meant to keep them alive. So when a nurse comes in, some of you will be nurses, and they get a patient sample, whether it's a sputum sample or a swab from their nose or a throat culture swab, that sample has to get to the lab. And so it has to go in some type of media that keeps it sterile. We don't want any outside contamination. And we want to keep the bacteria alive. If there's 10 bacteria at the back of your throat, and we want to keep those 10 bacteria alive so they get to the lab and they can grow up and then we can identify them. And so transport media is meant just to keep them alive. It gives them just minimal amount of nutrients so they're just happy enough to stay alive, but not happy enough that those 10 bacteria became 10 million bacteria and we think the patient has like a raging infection that needs to get admitted to the ER right away. Now, most bacteria can't get grown on all of these plates. We've got tons of plates, we've got tons of tubes, but not all bacteria can be grown on plates and tubes. And so some other ways that we can grow different organisms are using animals themselves. A unique one that we have not been able to figure out how to grow in the lab is the bacteria that causes syphilis. But the random thing, there is an animal that we can grow the bacteria in. And it's one of those, I'm like, give you like 100 extra credit points if you would ever guess the animal. You know, random factoid. An armadillo. An armadillo uniquely has the exact chemical makeup that the syphilis bacteria thrives on. Which is why, yes, if you ever have contact with armadillos, you actually are at an increased risk of picking up syphilis. But most people around here don't have contact with armadillos, and don't have to worry about it, but it is one way you can get syphilis. But we know that, and so if we have to grow this particular bacteria, we can use the animal to be able to grow the bacteria for us. And this usually happens for a lot of people that are studying the bacteria. Now, without using live animals, because even just using animals, armadillo or not, a ton of paperwork that you have to go through, which means it's going to be expensive. So it's only use when necessary. We can use some cells. Cells are not usually used for bacteria growth, but cell cultures are very commonly used when we're trying to grow viruses. Because viruses, for them to grow, because they can't replicate on their own, they have to get inside of a cell, and then they hijack that cell. We can grow the viruses in the cells. Mm-hmm. Yeah. Like if you had syphilis, you can, yes. Yep, and that's how they study them. But it doesn't seem to, nope, it doesn't seem to affect them whatsoever. So if you ever see a random armadillo, you would have it's not like, oh, it's got sores, I shouldn't touch it. Nope, you would have no idea. So cell cultures, they look similar. They're grown in these big plastic vials. There's usually a fluid in there that makes the cells happily grow. And we can put viruses in there, and they will grow inside those cells. We can also use something known as an enrichment culture, which just means that it's almost like taking one of these media, is we are giving a lot of a particular nutrient to an organism. If we know we are growing something like Staph aureus, and we know Staph aureus really likes something, we do have media that has like excessive amounts of that. That's going to make that particular organism as happy as possible. It's called enrichment culture. These two will be familiar from lab a couple minutes ago. A low oxygen culture, these are those microaerophilic organisms, and then an aerobic or a no oxygen culture. If we are working with organisms that are those, if you want a low oxygen environment, you use the candle jar. If you want a no oxygen environment, you use the gas pack. So this is showing when we crack open that little pack in there, it releases the hydrogen gas, which binds up with oxygen, and makes water, but it binds up all the oxygen. Now, if we grow our organisms, sometimes we want to keep them around for a while. There's a couple different ways we can preserve our organisms. One is that we can use a refrigeration, which is preserving our organisms for a short period of time. It's why we have a refrigerator up in our lab. So your plates went into the incubator before we left the lab. Arlene is going to pull them out tomorrow. So they just get incubated for 24 hours. That's enough time for them to just take all that media and just grow like crazy, and they will show all the colonies and get to their optimal size. And then we just pull them out because they have to sit somewhere until next Monday. And so we can put them in a refrigerator. That doesn't kill the bacteria. Refrigeration does not kill bacteria. It just slows their growth. So it's almost just putting a brake on their growth. So they're still alive. They're just chilling in the refrigerator. Short periods of time. We can't keep them in a refrigerator for months. They will eventually die. If we want to keep them for longer, we do have deep freezers. This is not your regular old freezer at your house. We have very specific freezers that go minus 80 degrees Celsius. We know minus 80 degrees Celsius is going to put those bacteria into like a zero growth state. It will literally make them go dormant. They are not going to grow. They're not going to die. If you put them in there long enough, I guess. In decades, they'll slowly start to die. But we know this is the temperature that will preserve these organisms. So when you've got a tube today, like E. coli or Staph aureus, Arlene had to grab a little sample of that from our minus 80 freezer and warm them up and make them happy. If we want to keep stuff for even longer than just years, we want to keep things around for decades, even centuries, we can use a process known as lyophilization. It's the fancy word for saying freeze drying. Is we remove the water and freeze it. The idea is that what happens to water when it freezes? It expands, and when you have a cell and there's water-based and you freeze it, it starts to get big and it could potentially burst. It could cause damage. Even sometimes the ice crystals alone can have jagged edges and cause damage to the cells. So we know that there could be damage to cells over a long period of time. So if we want to keep these things for decades or even centuries, like the CDC might want to keep for a lot of stock cultures, they freeze dry them. They remove the water first. Kind of just almost preserving the genetic material. Now, kind of switching gears a little bit is we make our organisms as happy as possible because we want them to grow, but the process of our organisms growing, because they don't just grow bigger, they're all the same size. They just grow in number. And growth of a microbial population, all of our bacteria grow by a process known as binary fission. We made them happy, and as they're super happy, they will start to duplicate themselves, their genetic material, some of their organelles. So you can see them, they're duplicating that one single chromosome. So now we've got two. We've got the duplicate amount of organelles, and there's not a lot to these cells. And then they just get cut in half and divide. And they're back to the exact same size. So the bacteria don't grow in size, they just grow in numbers till they become visible to us. So binary just means two, that's where the bi comes from. Fission means split. Because fusion means to go together, fission means to split. Now, the length of time it takes for them to go through that process is known as the generation time. It is also sometimes called the doubling time. You know, you had one bacteria, now you have two. You doubled. Or you had two bacteria to begin with. Two bacteria here, and they each go through binary fission. Now we have four. We doubled again. And they are constantly dividing in half, so they are constantly doubling. And the generation time is different for different bacteria and different factors. You know, how warm did you make them? Did you give them all the nutrients? It is not uncommon for bacteria to have a generation time between 30 and 60 minutes. Sometimes even less. If you make them warm and happy and give them lots of nutrients, these things are just going to keep growing and dividing every 30 to 60 minutes. This becomes an issue in the summer on a hot day at a picnic. If you have food that is sitting out, it's got nutrients, now it's warm, every 30 to 60 minutes, any bacteria that are in there are doubled. So if you had 100 in there, 30 minutes later, now there's 200. You know, another 30 minutes, now there's 400. Then there's 800, then 1600, and then so on. And it grows exponentially fast for the number of bacteria. So this is showing, you know, if you started off with one bacteria, after 30 minutes, it's 2. After another 30 minutes, we have 4. After another, we've got 8. After another, we've got 15. And it just keeps doubling. So if it's been sitting out for like 5 hours, what is that, like 12 generations? No. Generation times? That, I don't know, do the math. You're probably into the millions of bacteria at that point. Enough to cause food poisoning. So any bacteria, because of that doubling, grow in what's known as logarithmic growth. It starts slow, 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. But then all of a sudden it's like, oh, but that 16 became 32, and the 32 became 64, and then now we're up in the hundreds, and then in the thousands very, very quickly. That's what our bacteria do. Other organisms, have a nice slow, steady increase, like humans. We don't see this massive logarithmic growth, although if you look at hundreds of years we do. But, it's kind of a slow, steady increase. Not bacteria. Super fast growing. Now, as they grow, they do go through different phases. You do have to know these phases. There's a lag phase, a log phase, a stationary phase, and a death phase. Lag phase is if we put bacteria on plates, just like we did this morning, immediately they are not dividing. They're just like, oh, it's not so bad. Oh, here, let me break those red blood cells down because I need the nutrients that are in them. They're adjusting to their environment. They're starting to break down nutrients. They're just, doing everything in preparation to divide. But we don't actually have any growth here, so they call it a lag. They made their enzymes, and they're starting to break down all the nutrients that they need. That's when we go into that log phase or that exponential, very rapid growth. They are just doubling every 30 minutes. Now, it is at this point why we pull them out of the incubators at 24 hours. It's because at this point, they are so happily growing, and the majority of the bacteria are alive, and so they're going to gram stain accurately, and so they're good for gram staining. So we pull them out of the incubator at 24 hours. Put them in the fridge. Show there. Then enters what's known as the stationary phase. Well, these bacteria are breaking down lots of nutrients, but they're still living organisms, and just like we consume nutrients, we produce waste. The bacteria, as they break down nutrients, they produce waste, and that waste products that they make from the breakdown of these different chemicals can be deadly to bacteria. And so as you have bacteria growing, you have the exact same number as dying. And so the stationary phase doesn't mean nothing's growing. It just means the number of cells that are alive on your plate stays the same. If you got 100 new cells that just got born as they divided, you have 100 cells that just died. It does not make gram staining great because cells do not gram stain accurately if they're dead because they've already started to break down. The death phase just means you have more cells dying than are produced. So it still doesn't mean you don't have any new bacteria. You do. Bacteria are still growing and dividing, but they're dying faster than they're growing and dividing. So you are getting less live bacteria. Another picture out of the textbook that just shows that lag phase, exponential growth phase, happily growing, stationary phase. The bottom here, those orange is like the dead bacteria that sank. That as they're growing, you still have some dead cells that are forming, and then death phase where more are dying than they are growing. I think this picture came out of your textbook. This one I liked because it had all the descriptions underneath the thing. How we can measure whether bacteria grew and how many bacteria are present. There's a couple different ways, direct methods and indirect methods. And then some direct methods do not require incubation and some do. So a direct method of counting how many bacteria we have that doesn't require any incubation whatsoever, no counting on a plate, are two types. We have electronic things that will count our bacteria for us, and we have a way we can use microscopes to count our bacteria. So the two electronic counters, we don't have either in our lab that we use because we don't need to count bacteria. One is called a filter counter, down here. that just like you had that broth upstairs, we would take that broth, here's your little tube, and there's a device that we would drop right down into that tube, that broth. And again, you saw it was nice and cloudy. The more cloudy it is, the more bacteria there is. And this electronic device has a little gap in its electrode, and anytime a bacteria passes through that little tiny gap, a computer measures it. It's great. Now, it's going to give a really good estimate. Obviously, we're not like siphoning all the bacteria through, but it's going to give a really good estimate exactly how many bacteria are in that fluid. Another similar, the flow cytometry, is that as you allow bacteria to flow past this little laser, this laser will hit bacteria and it will send light refracting all through a machine, and that light refraction counts the bacteria. Again, it's different electronic ways we can just count how many bacteria there are. We don't do either, mostly because they're extremely expensive. Our way, if we ever had to do microscopic counts, is we wanted to actually know how many bacteria were in that fluid, and we wanted to know, per milliliter, is we do have microscope slides that on the slide, there is an etched grid. And it's such a small etched grid, you see it really well underneath the microscope. So it has a permanently etched grid, so this is not a disposable slide. And we can put a drop of that broth on the slide, right on top of that grid, and we put a cover slip on top of it. And so we know exactly, like if here's my slide, I put a drop right here, and then I put a cover slip that's going to flatten it. We'll say, here's my drop. Here's my cover slip. We know exactly how much fluid is in there. We can say, well, I know there's 0.1 or 0.1 milliliters between that cover slip and the slide. And then I can count the number of bacteria in a particular grid. And so I can say, oh, there's like 16 or 17, I think, little bacteria on this grid. And so I know 1 millimeter, I guess a tenth of that, times 1 millimeter times 1 millimeter of space can give us our cubic millimeters, or our volume. And we can say, oh, well, there's 17 bacteria per microliter of fluid. We can get a really good guesstimate just because we know how much fluid is in between that cover slip and the slide. So I'm like, I've done it before. It's kind of a pain. And as you notice, not every grid is identical. Some have fewer bacteria, some have more bacteria. The idea is that you count several grids, and it gives you a really good estimate of how many bacteria are in that fluid. Ways we can measure how much bacteria is present that requires incubation, which means more time, more money. One, we can do what's known as serial dilution. I could take that broth that I gave you that probably had 1 trillion bacteria, and I could get a whole bunch of other broth tubes, and I could dilute it out. I can take a little tiny bit and put it in the next tube, and then from that I take a little bit and put it into the next tube. That's this one. My clicker, I was going to say that here. So I could take that one broth that broth, that initial one that I'm at the top, initial original culture of 1 trillion bacteria. I can put it into a brand new tube, but I just do a 1 to 10 dilution. And even that, if I put it on a plate, way too many bacteria, you wouldn't be able to count the colonies. Or what if I did just 1/10 of that tube and put it into a brand new tube? Well, now I just diluted it 1 to 100, and there's still so many bacteria, it would be too numerous to count on a plate. So if I did another dilution, 1 to 1,000, now I'm at the point where I can start to count colonies. This one has 65. If I did another dilution, I'm down to 6, which, yes, 6 add another 0 is pretty close to 65. And so we're looking at how many colonies we have on that plate times our dilution. And that tells us how many actual bacteria are in that solution. We can say, oh, well, there's, if we do 65, then because we only put 1/10 of a mil, that's already times 10. 650 colonies times 1,000. 650 colonies times 1,000, 650,000 colonies per milliliter of fluid of bacteria. And we can bring a pretty good estimate, guesstimate. We don't know exactly, maybe it's 640,000. But it's a pretty good guesstimate on how many bacteria are present in that fluid. The problem with this, it's a lot of tubes and a lot of plates, and a lot of time. And so if they can find another way to save time and money, we will. I think these are my next slides. The membrane filtration. Membrane filtration is used when you know you have a very small amount of bacteria in a very large volume of fluid. So if we want to know how many bacteria there are per gallon or per 1,000 gallons, and we know there might only be one or two, that if I took a small sample, if I took a loop from your lab and picked up a little loopful out of that gallon jug of water, nothing's probably going to grow on there because maybe there's only 4 bacteria in the entire thing. So what they can do is they can take that large volume of fluid and they put it through a membrane. They use a vacuum to pull it through. But that membrane is going to catch any bacteria that are there. And then we set that membrane right on top of an agar plate to give it all the nutrients that it wants. And we let it incubate and grow. And then 24 hours later, we can see exactly how many bacteria there are. We can say, like, oh, there's like 10 bacteria out of that gallon of liquid. So this is used a lot, the DNR in the summer, if they want to know how many bacteria are present in the Mississippi. They just take, a big, huge gallon of it. They can run it through a membrane, and they can get the bacterial counts. This is exactly what the plates look like. That comes from the Mississippi. It's a fecal coliform bacteria. I know they are doing this now, testing for a particular virus. And so it's actually, they are siphoning it down in New York right now. Maybe you've seen it in the news. They are now discovering polio in wastewater in New York. That they are taking large volumes of wastewater, running it through filters, and trying to collect the genetic material left behind. And then whatever is on this membrane, they test it for that DNA that's found in the polio virus. I know they did this in the Mississippi. They were looking for a particular virus that affected fish, that they siphoned large volumes of fluid, and they are just looking for the DNA that gets caught up for that very specific organism. So we filter when we have large volumes of fluid, but expect little bits of organism. The last one is the most probable number. There are some bacteria we cannot grow on a plate. And so we can't just count plates. Instead, we can still do dilutions. And instead we take and add to the dilutions to the fluids. We add some type of substrate that if bacteria are present in enough quantity, we will see a color change. So this is for bacteria that just can't get grown on plates. And we do various dilutions, and as we notice, as we start to dilute the fluid, and we put it in 5 tubes each for each dilution, we notice that, okay, if we didn't dilute it, 4 out of the 5 tubes turn positive. As we start to dilute it, less of them turn positive. And we can start to look at all the dilutions and then look at the number of tubes that turn positive. And we've got charts that then tell us, well, based on if these dilutions and these tubes turn positive, this is about your quantity of bacteria. Another way we can make guesstimates of how much bacteria is present. Now, other ways that we can measure microbial growth that are indirect methods. One is metabolic activity. Again, if we start to see color changes, maybe a brighter color change indicates more presence of bacteria. Weigh the bacteria. If I wanted to, I could scrape all the bacteria right off the surface of that agar and weigh them. The more there are, the more they weigh. We also have genetic methods now, like PCR, where we can isolate the DNA and it can quantify it as well. Our easiest way for our lab, turbidity. The more cloudy it is, the more bacteria there are. We don't have an exact number of bacteria, but we do know the cloudier it is, the more bacteria that are present. There are certain machines that will tell us how turbid or cloudy something is. We don't use them. We just don't need to quantify it. But there are tubes that will shine light through the media. If there's nothing growing, the light can shine through it easily. If there's a whole bunch of bacteria, As the light hits the bacteria, it scatters and doesn't go directly through, and then it measures that as turbidity. Let's finish this PowerPoint, but I'm still going to start the next one. PowerPoint, what's due before I see you next time? There is a quiz. Due Sunday night. Should be open. Shannon, you said you were an advanced A&P. You got into metabolism yet? I know they usually go pretty hand in hand with each other. I don't know why this is done. I'm going to hand out one sheet again. I'm not collecting this. This is for you just to write down notes. Not sure how far we'll get into it today. Maybe I can just write all of it. It's great for just organizing notes. So some different things when we talk about metabolism. I think everyone at this point, you know, hate this kind of person when they say, I've got a super fast metabolism, can eat anything they want. The word metabolism just means all the chemical processes that are happening inside the body. Might be one of my first slides. So we're going to talk about, you know, what is metabolism in this PowerPoint? What are different types of chemical reactions? How do enzymes play a role? And then a large chunk of this PowerPoint, that's why I'm not sure how far we'll get to, is the breakdown of carbohydrates. And that's through the processes of the cellular cycle. And then right at the end of the PowerPoint, we talk about the opposite. Instead of breaking stuff down like carbohydrates, what are some processes that build things up, which is known as anabolism? And then how can we regulate some of this? A large part of the PowerPoint is the carbohydrate metabolism. So just the definition of metabolism is the collection of all the controlled biochemical reactions that take place within a microbe. So just all the chemical reactions that take place. There's lots of breaking things down. There's lots of building things up. Those are just chemical reactions that are taking place. If someone says they have a very fast metabolism, it just means all that breaking things down, building things up, happen very quickly and usually a little more efficiently. All it means. All the chemical reactions that are taking place. Now, the ultimate function of metabolism, of breaking stuff down and building things up, if you're a bacteria, is just to reproduce. They have to break down nutrients that they need so that they can just grow and divide. Now, of all the different types of metabolic reactions, we group them into two types: catabolic pathways or reactions, anabolic pathways. Anything that has that word catabolic or metabolism means breaking down. My way for remembering that, if you have a cat or know anyone that has a cat, cats break things. They knock things over. They knock a vase over. They break things all the time. So cats break things down. It's like dying as ever. Anabolic, then, is the opposite. Building things. Building larger things. And if my cat knocks a vase over, that's a catabolic reaction. That vase just scattered into like 100 different pieces, broke down. That's metabolism. If, however, I want to take those 100 pieces and put them together like a big old jigsaw puzzle, and I wanted to put that base back together as is, that would be an anabolic process, as I'm taking all the small little pieces and I'm building something larger. Which one takes more energy? To break the vase or to build the vase? Build. So anabolic reactions always require energy, so building something, where catabolic reactions release energy. It's a lot of effort to actually put the base back together. The picture up here, which I think I've got a bigger version on my next slide, catabolic reactions release energy, ATP. Anabolic reactions use that ATP to build the larger molecules. Now, there are 8 elementary statements of metabolism. So I'm going to have like my 1, 2, 3, 4, 5, 6, 7, 8. Just a couple of notes, and then puts them on top of my little diagram. 1, that all organisms require nutrients. Nothing's going to live if it doesn't have nutrients. So all organisms, if this is one big old cell, requires nutrients. The second, my little number 2 that's on top of the catabolism, metabolism gains energy from catabolism. So metabolism, like all the chemical process, gets its energy from catabolism. That breaking down releases the energy that's needed. If I'm not talking bacteria, I'm even talking us. If the nutrient you ate this morning for breakfast is, I had some toast. Well, you ate that toast, your digestive system is breaking it down and it's releasing energy so that you can go about your day. So that all your muscles can do everything they need to and all your cells can do everything they need to. Now that energy that is released is in the form of ATP. So ATP is the stored energy because it doesn't always get used instantaneously. Catabolism, that breaking stuff down, does give us more than just some energy. It gives us what are called, right on top of it now, precursor molecules. If I ate some toast, there are carbohydrates in there. And those carbohydrates eventually get broken down into simple sugars. And those simple sugars can then, as I'm reading ahead, go into an anabolic process to build something different, something bigger. But it gives us our basic building blocks. If I put some peanut butter on it, well, now I've got some proteins and different amino acids too. And I've got all these different smaller things that I can then use to build whatever I need to build. And so in anabolism, the precursor molecules and the ATP, because you need energy, used to make larger molecules. We have precursor molecules and we have the energy. In my vase, I had all the pieces of the vase and I had the energy and motivation to do it. I can then make a larger molecule. From larger molecules, we get even larger molecules. It's literally larger building blocks to macromolecules. Macro meaning large. We can take sugars and put them in together of like larger carbohydrates, and then maybe we can take those and mix it with some proteins. And eventually we get these larger, more complex structures. Macromolecules. My 7 on the bottom, the cell grows with macromolecules. Again, our goal is, yes, we want to reproduce, but the cell is getting larger. It's taking all those macromolecules that it is now creating, and it isn't growing. It's going to divide. But then my number 8, binary fission. Is that organism became big enough that divided into 2. We have the binary figure. So the ultimate goal is taking nutrients, breaking it down, building things up, growing, and then dividing. A bacteria. Now, when we do break things down and build things up, I'm not going to place it. When we break things down and build things up, a lot of times as we break things down, we do release not just energy, but we release electrons. And electrons carry energy. Anything that has extra electrons does actually have extra energy with it. So as we break things down, things are releasing electrons. There's always something right there to grab that electron, because electrons don't just float around freely. And so we call the reactions where something's always losing an electron and then something's picking it up. They're called redox reactions or reduction-oxidation reactions. Anything that picks up the electron, I was going to say, becomes the acceptor, the thing that's giving away the electrons, called the donor. And they happen simultaneously because electrons aren't floating around hanging out. As soon as something releases an electron, something picks it up. So on my 2 molecules, they're hanging out together. My molecule A is going to lose an electron. The molecule B is going to pick up that electron. Now, molecule A just lost an electron. And what charge are electrons? Positive or negative? They're negative. And so my molecule A lost an electron, negatively charged. It is now positively charged. I always say, because if you lose your negative friends, you become more positive. So it lost something negative, now it's more positive. Extra electron that A give it. And so it picked up something negative, so it is now more negative. The process of losing an electron is called oxidation. The process of picking up that electron is called reduction, which this sounds like an oxymoron. How do you gain something and also become reduced? God, science sucks. Well, you have to remember what you're picking up. So it's gaining an electron, but what charge is an electron? Negative. So you're gaining something negative. So your charge is becoming reduced. It's less. If you are gaining something negative, you become reduced. It's like you pick up a whole bunch of negative friends, you become reduced. That goes with it. Now my way, I didn't come up with it, but it's of remembering which process loses, which process gains, is if you can remember the acronym oil rig, you know, where they drill for oil. Oil oxidation is loss, reduction is gain. Stuff is breaking down and building up. There is moving around of a lot of electrons. Because even in the breaking down of carbohydrates, which we'll get to on Monday, there's a whole process called the electron transport chain. Electrons do carry energy. Now there are 3 molecules. In my last picture, you know, we had B picked up that extra electron. But there are a couple molecules that are just really good at picking up electrons. And so we call them electron carriers. Their job is really to go around, they're like the little taxi cabs of electrons, just picking up any extra electrons that it possibly can. In my note, when it picks up an electron, because an electron is a negative charge, it usually does try to pick up a hydrogen ion as well, try to neutralize that charge. And the 3 important electron carriers, no way would I ever know the names. I don't even think I could spell them or write them out either. I just want you familiar with their acronyms, because you will see these. So the 3 most important electron carriers are like NADH, NADPH, and FADH2. Now, before they picked up the electrons, this is what they were called. So this one picked up 2 electrons, and then it picked up 2 hydrogen ions to offset the charge. This one picked up 1 hydrogen, or 1 electron, and picked up a hydrogen. This one picked up another electron and picked up a hydrogen. That's why you see these have hydrogen on it, because they're carrying electrons, and those hydrogens just help neutralize it. So NADH, NADPH, and FADH2, I would recognize those as important electron carriers because they are going to come out of it. Now, in catabolic reactions, as we are starting to break things down, energy that we get is concentrated and stored in high energy bonds. So anywhere there's a high energy bond is where we're going to have all of that stored energy. And I'm going to give you an example down on the bottom. So here's your molecule of ATP. This stands for adenosine triphosphates because there are 3 phosphate groups. ATP is an energy carrier, where we store energy, and it's stored in these high energy bonds between the phosphate groups. If we want to release the energy in these high energy bonds, we just break a phosphate group off. And then ATP becomes ADP. Instead of adenosine triphosphate, it becomes adenosine diphosphate. We lost a phosphate. And it releases that energy in those high energy bonds. By storing energy in these high energy bonds in the phosphate, it's called phosphorylation. It's just having that energy stored in that high energy bond of phosphate groups. This is the most common molecule that we can easily add phosphate groups on and easily take them off when needed. High energy bonds. So if you ate, your breakfast, you're like, nope, I'm a big breakfast eater. You ate some pancakes and eggs and bacon and toast, and you had a huge breakfast. You probably didn't instantaneously use all that energy from that breakfast. Instead, your body broke it down, and it's going to store that energy in that breakfast for later today, and it does that by adding those extra phosphates on. At some point, you're going to have to make some molecules, and so I can use the energy that's in the ATP by breaking the high energy bonds. So first we're going to store the energy in the bonds, but then when we want the energy, we're just going to break the bonds. I'm going to end right there, because otherwise we start a whole nother section. Oh, there's my energy bond picture. We start a whole nother section on proteins and enzymes. And I'm not going to even get into it because we only have a couple minutes left. I didn't even get to the paper yet. That's how I'm like that I handed it out. Let's bring it back on Monday. Just the reminders before I see you Monday to take that quiz on Chapter 7. We open now, so you have to wait till Sunday night. And then we will, if we don't the PowerPoint Monday, we'll get pretty close, and whatever we don't finish Monday, I'll record it. Otherwise, I think that's it between now and Monday. Again, if you have any questions, send me an email. Just my note. I'll be off the grid on Thursday, Saturday night, Sunday morning, somewhere in there, trying to survive the cold. We'll see.